everyone. I hope you are all doing well and are adapting to our new online life for the class. I'm going to go to the next topic under Module 3, which is how cells reproduce. We just finished the microbiology. Prior to that, we talked about how cells make energy. And now we're going to talk about how cells make more of themselves. You might remember we talked about that everybody starts out as one cell, and then now you have trillions of cells. So cells clearly need a way of making copies of themselves. So that's what we're going to talk about in this presentation. I've divided it into two parts. So there's one called Mitosis Part 1, which is what we're doing today. And then there will be a quiz that is associated with this, a Canvas quiz that will be available for you to do after you do this part of the PowerPoint. And then we will do Mitosis Part 2. And then there will be another Canvas quiz related to the material for that. So let's get started. What we're going to talk about first is the process or the kind of the cycle of a cell's life. And I'm going to go to the picture versus the words on the slide before. And this is called the cell cycle. And what's a little bit confusing about it, because the cell cycle doesn't refer to the life cycle of a cell. It's not talking about from when the cell forms until it dies. It's talking about the period from when the cell first is formed until then it duplicates and forms two new cells. What we're talking about here are somatic cells. Let me change the color, that's not working. And somatic cells means body cells. So this process occurs to all cells except reproductive cells, which will be sperm and egg cells. We'll talk next in Module 4 about how reproductive cells reproduce, and that's by a slightly different process. So what we're talking about today is somatic cells, which are all your cells except for sperm and egg cells. And so this is how you grow. So you start off as one cell, and then you go through this cell cycle and you make new cells. So we're starting here with this cell here is a brand new cell that was just formed. And so it enters into what's called the cell cycle. And the cell cycle is a period of stages that end with the cell dividing and forming two new cells. So the cell cycle has 4 parts. The first one is called G1. The second is S, G2, and M. And so we're going to go through each of these in detail. The G1, S, and G2 together make up what's called interphase. And then M is when the cell actually divides. And so G1, S, and G2 are the most of the cell's life. It's the time when it's not dividing to make a new cell. So I'm going to talk about each of these phases separately. The first one is G1, and this is the longest phase. And G1 is when the new cell forms, it fully develops and grows, and then it does whatever that kind of cell does. So if it's a stomach cell, it does stomach things. If it's a brain cell, it does brain things. If it's a skin cell, it does skin things. So whatever that cell does, that's when it's functioning. It's during the G1 phase. The G1 is long, and some cells, they never really leave G1. They spend their whole life in G1. But at some point for many cells, they get a signal to divide. And so when they get that signal to divide, they leave G1 and enter the S phase. And if you think about it, if a cell is going to divide to be 2 cells, it has a set of its DNA, but it needs another set to have 2 sets, one for each of the new cells. New cells are called daughter cells, not son cells. So during the S phase, what the cell does is make a copy of all of its DNA so that it'll have a copy for each of the daughter cells. So that happens during S phase. When that's completed, the cell enters what's called G2 phase, and that's kind of the getting ready to divide stage. So during that time, the cell kind of gets organized. If you think about it, if a cell is going to split into 2 new cells, each of those cells have to have mitochondria and endoplasmic reticulum and Golgi apparatus, and that has to all be kind of divvied up. So that occurs during G2. When that's completed, the cell then enters M phase, and during M phase is when the cell actually divides. So the process of division is called mitosis, and we're going to get into that in more detail. So that's what the cell cycle is. The first 3 parts are called interphase, which is G1, S, and G2. When that's complete, the cell enters the M phase, which is where it actually divides. Oops, excuse me. So this is just another picture which kind of shows the same thing. So here we have our brand new cell. It enters into G1, which is primarily growth. Then if it gets the signal, it leaves G1, enters into the S phase, which is when it makes a copy of all of its DNA. And then it enters into G2, which it prepares for division. And then it enters M phase or mitotic phase, and that's where it actually divides. So interphase is everything but the mitotic phase. It's G1, S, and G2. And then the mitotic phase is the actual cell division. Okay, again, this kind of summarizes the 3 parts of interphase. So interphase is most of the life of a cell. Most of its life, the cell is in interphase, and that's when it does its normal functions of whatever type cell it is. It varies with different types of cells. For example, your skin cells don't live that long, and so they reproduce pretty quickly, whereas some cells, like your brain cells, unfortunately, almost never divide, and so they never leave G1 but stay in G1 for a long period of time. So it varies according to cell type, how quickly cells divide. And again, G1, this is like a summary of what we already talked about. Cell grows. S phase is when it makes a copy of its DNA. G2 is when it gets ready to divide. That brings us to mitosis, or the M phase, mitotic phase. They all mean the same thing. And the M phase of the cell cycle has 2 parts. And usually they're kind of considered together. Mitosis is the actual division of the nucleus. So that's taking 1 nuclei and forming 2 nuclei. Cytokinesis is the splitting of the cell with the 2 nuclei in it into 2 new cells, each having a nucleus. So mitosis is division of the nucleus, which is what nuclear division means, and cytokinesis is division of the whole cell into 2 cells. So those are the 2 parts of the M stage. So at the end of mitosis, you have 2 daughter cells that are genetically the same. so they each have the exact same sets of genes. They're exactly like the parent cell that's split, so that every time you make new skin cells, for example, those new skin cells are just like your old skin cells. So next, we're going to primarily talk about mitosis. And before we talk about mitosis, we have to talk a little bit about how our DNA is organized inside of our nucleus. DNA is a very long molecule, and when a cell is not dividing, it's kind of a loose threads of DNA kind of all intertangled inside of your nucleus. I'll show you a picture in a minute. However, when the cell is undergoing mitosis, remember that's division of the nucleus, that DNA gets organized into something called chromosomes, okay? And each species has a characteristic number of chromosomes. We have 46 chromosomes. So every human cell, except for sperm and egg cells, have 46 chromosomes, whether it's a skin cell or a stomach cell. All of those have the same sets of chromosomes. Those chromosomes aren't really visible as distinct chromosomes, except when a cell is undergoing mitosis. So this is kind of showing that. So this picture here, the purple is the nuclear envelope. So this is the nucleus of your cell. And when the cell is not dividing, the DNA, I kind of call this a bowl of spaghetti version because it's just these tangled threads inside. And each of those threads is a highly coiled piece of DNA. DNA has this double helix structure, and it twists and twists around these proteins. It's kind of like if you take a piece of yarn or string and just keep twisting it and twisting it, it gets all coiled. And so that's what makes up these strands or threads of DNA. They're not just one strand of DNA, but it's these highly coiled strands of DNA. And the proteins it's wrapped around that kind of form a base are called histones. So that's the way your DNA is actually in your cell when your cell is not dividing. When it goes to divide, that DNA becomes organized into something called a chromosome. And this is a picture of a chromosome. And this is actually a duplicated chromosome, because remember during the S phase, if you remember, you make a copy of all of your DNA. So this would have been one original chromosome. And then this is the copy that you made during the S phase. So you actually have two identical copies of every chromosome as it enters into mitosis. They are joined or hooked together at something called the centromere, which is this region right here, is where they're joined together. So in a human cell, you have 46 chromosomes, and then right before mitosis begins, each of them has a copy. So technically, at that time, if you count the two chromosomes, you have 92 chromosomes because you have a copy of every one of your 46 chromosomes. These chromosomes that are hooked together are called sister chromatids. That's on another slide. You'll see it again. And that's what they're called when they're still hooked together like that, sister chromatids. This is just a picture of this is actually a plant cell. And so this is when the nucleus is in kind of that bowl of spaghetti formation. And this is where the chromosomes are visible. You can kind of see these thicker coils visible inside of the cell. And so that cell is getting ready to, or has entered into mitosis, is getting ready to divide. This is just an example. The number of chromosomes is just the way a species organizes its DNA. It doesn't really mean anything in terms of how big the organism or how complex it is. We have 46 chromosomes. A goldfish has 94 chromosomes, but it's a number specific for that species. We'll be talking a lot more about chromosomes when we get to genetics, but this just gives you an example. So we have 46 chromosomes, right? Which is here, but we actually have 23 pairs because when we're formed as a fertilized egg, the egg has 23 chromosomes and the sperm has 23 chromosomes. So they come together and form the embryo, which now has 46. So we have one set of chromosomes from our mother and another set from the father. And so those are pairs. They contain same sets of genes. And so they come together to give us our 46. So cells that have 2 of every chromosome, that is, they have chromosome pairs, are called diploid. So all of our cells, except for sperm and egg cells, are diploid. And we have 23 pairs, and those pairs are originally from our parents. So we have one set that came from our mother, one set that came from our father. That pair of chromosomes are also called homologous, which is this word here. So this is a homologous pair of chromosomes. One was from your mother and one was from your father. And each chromosome has the same set of genes. They could have different versions, like your father could have the eye color gene could be blue and your mother's could be brown. We'll talk more about that when we get to genetics. But that's what your 46 chromosomes are. They represent 23. Now, what gets confusing is that when the chromosomes duplicate during the S phase, we make a copy of every one of those 46 chromosomes. And as I mentioned, that's called our sister chromatids. And they are attached at the centromere. So let me show you that. So here is the original 1 chromosome from your dad, say, and this was your 1 chromosome from your mom. The colors don't really mean anything. And then during the S phase, they make a copy. So this is your 1 from your mom, and you made an exact copy. They're joined at the centromere, which is this thing right here. And so now you have that chromosome and its copy. This is the 1 from your dad. During the S phase, you make a copy of that one too. And they're attached. Those are the sister chromatids. So these are sisters and these are sisters. Okay. And then the 1 blue and the 1 pink are homologous chromosomes. They represent the original pair from each parent, 1 from each parent. So technically, right before mitosis, you actually have 4 of every chromosome. You have your homologous pair, which will be 1 blue and 1 pink, and then each of them have made a copy. So you have 2 blue and 2 pink. This gets kind of confusing. This is just a picture of a real chromosome that has been duplicated. So these are the sister chromatids, the 2, and they're joined together at a centromere is where they're hooked together. So mitosis is the process of making somatic cells, that is all cells except for sperm and egg cells. And that's the process that's used for all your growth and cell replacement. So from the time you were a fertilized egg of 1 cell to your current state where you have trillions of cells, you got from 1 to trillions by mitosis. Also, if you repair or grow things, like if you when you make new skin cells, you make new blood cells. If you damage a tissue, like you get a cut and you have to make new cells to replace them, that's all done by mitosis. And that's what we're going to talk about in detail. The other process is meiosis, which is the mechanism for making sperm and egg cells. And we're going to talk about that separately a little bit later. And this is just a picture so that from the time of fertilization, you have cells that only have one set, and then you form a fertilized egg, and all of the growth from the fertilized egg to adulthood is done by mitosis.